influenza. Okay. By the way, the influenza viruses are not the only orthomyxoviruses, okay? The other genus under Orthomyxoviridae is genus Thogotovirus, but it is not known to cause disease to humans. Okay, so there are three immunologic types of influenza that are known to affect humans. Okay, so they are designated A, B, and C. So here we are again, no, with our medical creativity. There is actually a fourth type, no, type D, but it is not yet known to affect humans. So only A, B, and C involve humans. All right. So the influenza viruses are generally RNA viruses. They are single stranded and they are in the negative sense, meaning before it can replicate, it has to be transcribed into a positive sense RNA first, no? By the enzyme RNA polymerase. So remember, the positive sense RNAs are the ones that can act as mRNA, no? And only then can it be translated to protein, right? So from negative sense, it has first to be transcribed to positive sense, right? And the influenza viruses or the influenza virus is a multipartite virus, okay? Meaning it has segmented nucleic acid genomes with 7 or 8 different RNA molecule segments. Each is coated by capsid proteins and forming helical nucleocapsids. Okay, so types A and B have 8 segments, and type C has 7. So later on, we will see what gene is lacking sa type C. Right. So each segment actually has an assigned protein to code for and different functions. So if you look at the table in your book, you will find the different segments, no? And you will see what proteins they code for. Okay. There are 9 types of structural proteins in each viral particle. Okay, wait. Let's go back to the segments. Let's draw each segment in slightly more detail. Okay, slightly lang ha. So just so we understand what's going on. Okay, so we have a protein called nucleoprotein or NP. Okay, this associates with the viral RNA to form a ribonucleic protein complex or RNP, okay, to which the RNA coils into, forming a helix. So that's why it's called a helical configuration. Okay, so this is the helical nucleocapsid I mentioned earlier. At the end of the nucleocapsid, you will find a complex of 3 large proteins. So again, using our creativity, let's name them protein A or PA and protein B. The third one is actually also a protein B, so let's name them B1 and B2. Okay, so we have PA, PB1, and PB2. So these 3 proteins are important for replication because this complex is actually the RNA polymerase complex. Okay, and this is going to be responsible for converting the negative sense RNA into the positive sense strands so that they can be translated to proteins later, okay, in the host cell. Now, this is only one segment. Remember, there are multiple segments present in an influenza virion. Okay, so each segment is assigned to code for a different protein, which you will see in the table I told you earlier. If we lay them all on the table, strand by strand, you will see that segment 1 codes for PB2, segment 2 codes for PB1, and segment 3 codes for PA. So these 3 will code for the 3 proteins that will be attached to the new RNA strands later, no, during viral replication. Segment 4 codes for hemagglutinin, 5 codes for the nucleoproteins that will associate with the new RNA strands later. Number 6 will code for neuraminidase, 7 codes for the M protein, And number 8 codes for a non-structural protein that functions to inhibit splicing of the negative strands. So, non-structural. So, let's call it NS. Actually, NS gid ang tawag saya guys ha. NS gid siya. Okay, so we mentioned that type A and type B have these 8 segments, but type C only has 7 because type C doesn't have a segment to code for neuraminidase, okay, or segment number 6. So this is what is lacking sa mga type C. So the M protein, there are two actually. M1 is the matrix protein which forms a shell around the segments underneath the envelope. It makes up around 40% of the viral protein. M2 is an ion channel protein that spans the viral envelope along with the NS2 protein. Remember that the influenza virus is an enveloped virus. Okay, so it has a lipid envelope that it actually derived from a host cell. On the surface of the envelope, we will find spikes of two different types of glycoproteins, hemagglutinin and neuraminidase, or HA and NA, or many times H and N. For this lecture, when I mention H or N, I am referring to hemagglutinin and neuraminidase proteins, okay? Okay. So hemagglutinin makes up about 25% of viral protein and neuraminidase about 5% only. So mas daghan si hemagglutinin. Now, how do we distinguish between the different types of influenza viruses ba? Type A, type B, or type C? Is it by the hemagglutinin and neuraminidase proteins? Actually, no. It's actually based on the antigenic differences exhibited by two of the internal structural proteins, no? Nucleoprotein and the matrix proteins, not H or N. The differences between the H and N are actually used to subtype the type A viruses. Okay? Now, the standard nomenclature system for influenza viruses is the type. So again, based on the type of the nuclear material, particularly the NP and the M proteins. So whether it is type A or B or C. And then the host of origin. So this is placed if the original host is not a human. Okay? For human strains though, this is not placed anymore. Like for example, for type B and C. And then the place of origin or the location where it was first found, the strain number, and the year of isolation. Okay, and then for type A, we add parentheses subtype H and N. So for example, a type A H1N1 flu virus of duck origin from Alberta, California, 35th strain found in 1976 would be named A. Duck, Alberta, 3576 H1N1. Or a type A H1N1 flu virus of swine origin from Iowa, 15th strain found in 1930 would be A. Swine, Iowa, 1530 H1N1. Okay? But for strains from humans, just remove the animal host of origin. For example, A, Hong Kong, 368 H3N2.